Hello everyone, my name is Mike Schneider, and today my group members and I will be talking about implantable polymer materials for medical applications. So to give you a brief outline first, I will discuss what an implantable material is and some of the history behind them. Next, Troy will talk about the requirements of a scaffold and biomaterials used to make the scaffolds. Then Steph will talk about the structure of scaffolds and some of their functions. And finally, Tyler will cover some challenges that people in the industry have and some of the future uses and applications of them. So you might be wondering, what is an implantable material? Well, an implantable material is any material inserted or grafted into the body for prosthetic, therapeutic, diagnostic, or experimental purposes. It can be any material, but this presentation will focus primarily on polymers. Most of these materials are inserted into the body via surgery, and different types of implantable materials are let in the body for varying amounts of time, and some biodegradable polymers disappear without the need of a second surgery. Currently, polymers have a very widespread use in the medical field, but we will focus primarily on some more prevalent uses such as scaffolds. So a little bit of the history. Prior to the 1980s, implantable materials were not made of polymers, and traditional materials such as metals, ceramics, and in some cases glass were used. On the left is a metal-on-metal hip, and the right depicts titanium pins used for hand surgery. The reason for the switch away from traditional materials is because some polymers have several advantages, including weight, biocompatibility, and cost. One of the reasons there has been a surge recently in the use of polymers in implantable materials is because of concerns in 2012 regarding the wear and corrosion of metal-on-metal hip implants. These are some of the commonly used polymers in medical applications, and some of the notable ones include polyamide, which is used for tendon and ligament repair, polyvinyl fluoride, used for suture material or surgical mesh, and polymethyl acrylate used for bone cement.
There are 5 key components to consider when thinking about how to design a polymer scaffold: biocompatibility, biodegradability, mechanical properties, scaffold architecture, and manufacturing technology. The first and most basic criterion of a polymer scaffold is that the implanted material must be biocompatible with the host body. At a most basic level, that means that the host body cannot reject the implanted material. A negative immune response, such as severe inflammation, could actually reduce healing of the affected area or even bring about a worse health concerns, such as infection. According to an article written by Fergal O'Brien in Materials Today, the definition of biocompatible in reference to polymer scaffolds means that for an implantable material, cells must adhere, function normally, and migrate onto the surface and eventually through the scaffold and begin to proliferate before laying down new matrix. The second requirement is reliable biodegradability. Since polymer scaffolds are not permanent implants, one of the main objectives with their design is to allow the host body's own cells to eventually replace the implanted scaffold over a period of time. The body's cells must be able to produce their own extracellular matrix as the implanted polymer scaffold degrades away. With biodegradation comes byproducts, so another thing to think about is what byproducts will be produced with the degradation of the scaffold. These byproducts must not be toxic and must be able to travel out of the body without negatively interacting with any of the body's other organs or systems. Because tissue formation occurs simultaneously with scaffold degradation, a controlled inflammatory response along with continuous infusion of cells such as macrophages is required. The third important consideration of a polymer scaffold are its mechanical properties. For implantable materials, mechanical properties refer to a polymer scaffold's strength and ability to function properly in the anatomical area in which it was implanted. During the early stages, this means that it must be strong enough to handle surgical implantation and handling before it is actually in the body. After the scaffold is in the body, this means that it must be able to handle the stresses and forces that it will be subjected to during its life in the area of the body in which it was placed. For example, scaffolds for orthopedic applications, such as in the knees or shoulders, will experience the highest amount of stress because they are in weight-bearing areas of the body and must be strong enough to function from the time of implantation to the completion of the remodeling process. The fourth key component is scaffold architecture, and it is arguably the most important component of the five. This property refers to how a polymer scaffold structure is assembled. Ideally, the implanted material should have an interconnected pore structure with high porosity to allow for good cellular penetration and diffusion of materials in and out of the polymer construct. For the diffusion aspect, a highly porous structure allows cells to move into the structure to begin assembling the new extracellular membrane, and also for nutrients to travel to these cells within the structure. This also means that the waste products of biodegradation of the polymer structure are able to pass right out of the structure without being trapped or interfering with any nearby organs. The fifth component to consider is manufacturing technology. This category refers to a polymer scaffold's ability to become clinically and commercially viable. An ideal implantable scaffold should be cost-effective to produce and should also be possible to scale up from simple lab production to small batch production. There is not much else to this category, but it is important to consider how a certain scaffold is prepared and manufactured for clinical use compared to another scaffold designed for the same use. A final category to consider when designing a polymer scaffold is the type of implantable biomaterial to use. According to an article written by O'Brien in 1976, the medical world defined a biomaterial as a non-viable material used in a medical device intended to interact with biological systems. However, since then, the definition has changed a bit to a material intended to interface with biological systems to evaluate, treat, augment, or replace any tissue, organ, or function of the body. This change in definition reflects the shift in the goal of implantable biomaterials towards tissue regeneration. Typically, three types of biomaterials are used for the synthesis of scaffolds: ceramics, synthetic polymers, and natural polymers. Ceramic scaffolds such as hydroxyapatite, HA, and tricalcium phosphate, TCP, have been used for bone regeneration applications. Rather than being used for soft tissue regeneration like synthetic or natural polymers, ceramics are used for bone regeneration because of their high mechanical stiffness, low elasticity, and hard brittle surface. Additionally, the biocompatibility of ceramic scaffolds with bone applications is very high due to the similarity of chemical makeup and structure to the mineral phase of native bone. The next category of biomaterials is synthetic polymers. Some polymers that are typically used for the synthesis of synthetic polymers include polystyrene, polylactic acid, polyglycolic acid, and polylactic glycolic acid. The advantages of using synthetic polymers include being able to build a specific scaffold architecture and also being able to control the degradation rate by varying the polymer itself or the composition of the individual polymer. However, because these scaffolds are made of synthetic materials not commonly found in the body, a drawback is that they pose a significant risk of rejection by the host body. The third category of implantable materials is natural polymers, which are mainly used for tissue engineering. Some of these natural materials include collagen, various proteoglycans, alginate-based substrates, and chitosan. The advantage of using these materials is that they are extremely bioactive, so they promote greater cell adhesion and growth than synthetic polymers or ceramics. Additionally, they are highly degradable and allow the body's cells to properly and efficiently form their own extracellular matrix as their implanted scaffold degrades away. However, one drawback is that these natural polymers usually do not have strong mechanical properties, making them poor choices for orthopedic applications. Because all three of these categories have their own advantages and disadvantages, it has become more common in recent years to use a combination of the three in composite scaffolds to try and get the best properties from the three groups in a single implantable material.
You have already heard about some of the important polymers used in implantable material and the history behind them and their synthesis. I will continue from here, expanding into the application of many of these polymers in implantable materials. In the medical field, scaffolds are incredibly important. The scaffold is mostly used in tissue engineering for the following purposes: to allow a cell to migrate and attach to other cells, to preserve biochemical factors, to support the diffusion of vital nutrients and products, in an attempt to help fix cell damage due to an accident, infection, or other medical issues. To achieve this goal, scaffolds are three-dimensional biopolymer structures resembling the picture below. A cell is added and grows through the scaffold to achieve tissue regeneration. This can be done ex vivo or in vivo to allow for production of different types of tissue. Ex vivo is used to produce cell cultures outside of the body, as can be seen on the picture to the right of an ear. The scaffold was placed in a dish and the cells were added to grow to the required shape and size determined by the scaffold. It can then be added to the body, but has had limited use so far on humans. In vivo is used to describe scaffolds inserted into the body. As Troy has stated, they are usually biodegradable polymers to ensure the scaffold is removed after completing its goal. Non-degradable polymers are also used, but some are believed to have some dangerous side effects after long periods of time. Scaffolds are currently commercially produced for drug and medical treatment testing. There are many companies in this field, such as Pure Matrix, which was founded at MIT, that uses an array of different polymers to produce their scaffolds. Some of these common polymers have already been discussed, such as polyglycolic acid, polycaprolactone, and polylactic acid. Pure Matrix focuses on producing scaffolds that can be used to grow many organs for drug testing. The picture on the right is a scaffold in the shape of a heart, and after some time you can see that the cells slowly grew, filling in the scaffold, and eventually produced the final product, which was a small rat heart that can then be injected with different drugs, and the impact can be more easily seen as the heart is affected by the chemicals rather than testing on animals that shield the heart and must be removed later to see the effects. This expands also into real-world applications of scaffolds in humans. Many of the current scaffolds being produced are still under clinical testing, but have had limited tests on humans. Currently, these scaffolds have been used to produce bone grafts and cartilage that can then be inserted inside the body or can be used in vivo and produce bone inside the body. As can be seen in this slide, a scaffold was added in the yellow circle on the first image A. An X-ray after 4 weeks indicates some bone had regrown and was beginning to repair the defect. And by the 8th week, it had successfully recovered most of the bone loss. This was a common mutation found in humans and was tested on 18 people to find the effects of scaffolds. Also, bone grafts and cartilage produced ex vivo can be inserted into the body to repair things such as damaged knees where the cartilage has been lost. Outside of this, these scaffolds have also been used to produce different human organs, but are still experimental, with very few actual implants. But research were able to take cells from donated kidneys and produce replicas that could also have been used and inserted in humans, since there's currently a large missing amount of implantable kidneys from donors. My fellow group member Tyler will now discuss the challenges with implantable materials and future uses.
One of the largest challenges facing scaffolding is developing mechanically strong porous scaffolds that retain proper vascularization and the integration of the new engineered tissue with the host tissue. The reason for this large focus in vascularization is that there were incidents with the BTE, or bone tissue engineering, and skin tissue engineering, where the regeneration of the tissue was limited to only the edge of the scaffold upon implementation. This caused an improper generation of the tissue and was found to be a direct cause of the improper vascularization of the construct. Another big challenge is being able to incorporate strategies that modify or regulate immune functions that don't inhibit optimal tissue regeneration and integration. Also, surprisingly enough, one of the other challenges that bone tissue scaffolds face is healthcare burdens due to high manufacturing costs and its patient-specific design. Lastly, the fragile nature of proteins has motivated design of scaffolds that release DNA-containing genes that encode growth factor. Controlling the diffusion rates of these genes and proteins from scaffolds so that they're in the physiological range is another challenge researchers face. With other available bioabsorbable polymers such as polylactic acid, polyglycolic acid, and polydioxananode, they face challenges like releasing acids and other toxins through the dissolving and degrading process that occur in the body. For sutures and small staples devices, this is not a serious problem, because the quantity of harmful substances released is small, but for large devices such as weight-bearing screws, The release of harmful products can be significant and cause inflammation problems, similar to the problems that occurred with crude metal devices of the past. Also, they are soft and can deform with time, and due to the intensive interactions with the body, they can leach, leading to wear and tear. They also can absorb important nutrients and water from the blood. The challenges faced with scaffold design and fabrication leads to new opportunities for new and thrilling applications in research. This includes polymer assembly, chemical cues, improvement in biocompatibility, improvement in biodegradability, improvement in mechanical properties, and induced formation of natural tissue. Also, there are researchers attempting to tackle down the vascularization problem and the inhibition of fibrous tissue formation. One of the ideas in place is to engineer microvasculature by cells in the scaffolds prior to implementation. Another idea is incorporating growth factors by genetically modified cells or even by coating the scaffold with anti-inflammatory molecules. Other future products include nanotechnology, which can provide strategies that can help to create features on a scaffold in a dimensional range that may be adequate for cells and biomolecules. There have also been developments in efficient cell seeding methods to minimize in vitro culture of the BTE constructs and allow for maximized bone tissue regeneration in the in vivo. With other polymer implants, research has been going on to improve the tyrosine-based polymer technology as a platform technology with broad applications in orthopedics, cartilage and ligament repair, wound care, cardiovascular repair, and drug delivery. The outlook for continued development and commercialization of these technologies is excellent. In addition to this, there have been an emergence of combination polymers, which research believe to hold promise for the creation of novel materials that possess desired properties for highly specific applications. Furthermore, the development of processing techniques, especially with the assistance of computer-aided technology, is allowing for the formation of particles and scaffolds that, with extremely complex architectures that can mimic their biological counterparts.
So in conclusion, today we talked about what an implantable material is and some of the history behind them. We discussed the requirements of a scaffold and the biomaterials used to make the scaffold, as well as the structure of the scaffolds and some of their functions. We also discussed some of the challenges and future uses of the implantable materials. Hopefully now you are more aware of how implantable materials play a role in the medical field and why it is such a large, fast-growing field. Thank you.